Hello, my name is Sarah Finnegan and I am in Group 2B for Tissue Engineering. The title of our poster is The Determination of Mechanical Properties, Degradation Rates and Cellular Viability of Gelatin Beta TCP Scaffolds. Gelatin is a natural biopolymer derived from collagen. It is commonly utilised with beta-tricalcium phosphate to produce a nanocomposite scaffold. This study aims to assess differential characteristics between pristine gelatin and gelatin beta TCP scaffolds to determine the suitability for bone tissue engineering strategies by assessing mechanical properties, degradation rates, behaviour and cellular viability. Our hypothesis states the addition of beta-tricalcium to gelatin scaffolds seeded with pre-osteoblast cells creates a biopolymer scaffold with unchanged mechanical and biological properties. In order to carry out the experiment, we had to prepare our gelatin beta-tricalcium scaffolds using freeze-drying fabrication. We then carried out tensile testing on the rehydrated scaffolds to determine the compressive strength. We assessed the swellability by hydrating the scaffolds with ethanol and PBS, then incubated them for 7 days. Following this, we reweighed them to determine the degradation rates. The scaffolds were then seeded with MC3T3E1 pre-osteoblast cells, and their viability was tested using Hoist DNA assay and were quantified by measuring fluorescent intensity. In terms of results, the addition of TCP to gelatin scaffolds increased resistance of the scaffolds to elastic deformation under load. This is illustrated by a higher Young's modulus, as seen in figure 4 and 5. TCP has a marginally higher degradation rate, although both demonstrate higher rates. As seen in figure 6, both gelatin and TCP demonstrates high degradation rates, with TCP displaying 100% degradation and gelatin displaying 96% after 7 days incubation. From the standard reference curve, the known DNA concentration from the seeded cells were demonstrated at 485 nanometers. The concentrations were then converted to cell number, which is representative of cell viability for TCP and gelatin scaffolds at 1:10 and 1:20 dilutions, respectively. The gelatin TCP has a greater cellular viability than the gelatin. Upon statistical analysis of the results, we obtained a p-value of less than 0.05, which allowed us to reject our null hypothesis. This may imply that beta TCP alters mechanical and biological properties of a scaffold. However, a large sample size will be required for a more conclusive result.